This tutorial will provide more information about the Structure-Function Linkage Database, or SFLD, which links enzyme sequence and structural information to specific functional information. If you aren't already familiar with the organization of the SFLD, you may want to start with Part 1 of this tutorial series, the SFLD Overview. In this tutorial, I'll show you how you might use the SFLD to make some hypotheses about the function of an uncharacterized enzyme from the radical SAM superfamily, a diverse group of evolutionarily related enzymes that use a conserved 3-cysteine motif to bind an iron-sulfur cluster and activate S-adenosylmethionine. For this scenario, I'll start with an enzyme that has no associated experimental information and show you how you could use the SFLD to make some hypotheses about what its function might be. Here's an enzyme that's annotated in NCBI's GenBank database as an L-lysine 2,3 aminomutase. But if you look at the references, they're all large-scale sequencing papers, so the annotation doesn't seem to be based on specific experimental data. I'm going to get the amino acid sequence for this protein. I'm going to copy it, and back on the SFLD webpage, I'm going to choose the Search by Enzyme link at the top of the page. I'll then paste my sequence of interest into the protein sequence search box. There are two ways I can search the SFLD using a sequence. I can either do a blast search of all the sequences in the database, or I can search against the hidden Markov models in the database. Hidden Markov models are models based on sequence alignments, which give the probability of finding a particular residue in a particular column of the multiple alignment. The SFLD HMMs are based on our curated multiple sequence alignments for all the families, subgroups, and superfamilies in the database. I'm going to select the HMM search and press the search button. What I get back is a list of the groups that the sequence matches, with the best matches listed first. For each match, I have its level in the SFLD hierarchy, whether it's a family, subgroup, or superfamily. I also have the e-value and the score for the match. For family matches, the corresponding subgroup and superfamily are listed, and for subgroup matches, the corresponding superfamily is listed. As you can see, the top hit is the glutamate 2,3 aminomutase family. The L-lysine 2,3 aminomutase family, which the sequence was annotated to in the NCBI, is actually the third hit. The second hit is the PLP-dependent subgroup, which the glutamate 2,3 aminomutase family and lysine 2,3 aminomutase family are both part of. So just based on score, it looks like the sequence is more similar to the glutamate 2,3 aminomutase family than the lysine 2,3 aminomutase family. But let's look in a little more detail at how well the sequence fits into the two top family hits. I'm going to start by looking at the sequence in the context of the L-lysine 2,3 aminomutase family, because that's what it's annotated as in the NCBI database. I'll click this Align to Family link for L-lysine 2,3 aminomutase, and that takes me to a curated alignment for the L-lysine 2,3 aminomutase family. Scrolling down, you'll see that our sequence of interest has been added in at the bottom. The catalytic residues required for family-specific functionality are listed in this table at the bottom of the page. Each residue is associated with an evidence code that tells you what kind of information was used to determine its functional role. Mousing over the evidence code will give you a detailed explanation of what the code means. When applicable, residues are also associated with a literature reference icon, which is a clickable link. The catalytic residues are highlighted in the alignment, so it's easy to quickly scroll through and see whether your sequence of interest conserves the applicable catalytic residues. So I can see that though this sequence does have most of the catalytic residues, it has a lysine and asparagine in place of the 2 conserved aspartic acids required to bind the lysine substrate. So it looks like the GenBank annotation of L-lysine 2,3 aminomutase might be wrong. Let's go back to our HMM search results and look at this sequence in the context of the glutamate 2,3 aminomutase family to see if it fits into that family any better. Again, our sequence of interest has been added in at the bottom of the alignment. Scrolling through, I can see that in addition to good overall sequence similarity, this time our sequence of interest does in fact have all the family-specific catalytic residues we expect. So now that we've decided our sequence looks like a glutamate 2,3 aminomutase, let's get more information about the family. Scrolling back up to the top of the page, there's a link to the glutamate 2,3 aminomutase family. I click on that link, and that takes me to the family page. This page is organized similarly to the superfamily page you saw in part 1 of the tutorial series, so I'm not going to go into too much detail about that. At the top of the family page, you'll see summary information and an associated literature reference. Down at the bottom, you can see the overall reaction catalyzed by the family. If you want to look in more detail at the functional domains in the family, you can click on the link at the top of the page. You'll then see a table listing all of the functional domains in the family, as well as some associated annotation information, like species, and links to other databases. Clicking on the Toggle Columns tab will allow you to select which information is displayed in the table. Some things you might be interested in are operon context via Microbes Online, protein databank identifier, enzyme length, etc. You can also download this information as a tab delimited file, which you can open in any spreadsheet program like Excel, or you can download the sequence set. That concludes our brief tutorial on using SFLD search and alignment tools to make hypotheses regarding the function of an uncharacterized enzyme. For information on how you might use the SFLD, along with the Cytoscape Network Visualization Program, to visualize sequence relationships in large group of enzymes, check out Part 3 of this tutorial series.